So now we're going to talk about some different toxins in the human body. Some really fun ones that, well, not fun to have, but fun to discuss. And the first one that we're going to talk about is tetanus, which is actually a bacterial infection. And what tetanus will do is it'll result in skeletal muscle fibers basically contracting uncontrollably to the point at which death occurs, particularly from spasms over contraction of respiratory muscles, basically the inability to breathe. So how does tetanus cause these muscle contractions, these significant muscle spasms? To understand this, you have to understand that there are many neurons that synapse with alpha motor neurons in the spinal cord that are not just primary motor cortex neurons. Okay? There are other neurons in the spinal cord too that synapse with these alpha motor neurons, and the neurons that we're going to talk about in particular are called GABAergic neurons. Okay, so there are GABAergic neurons in the spinal cord that synapse with alpha motor neurons, and what exactly do they do? To understand this, you have to understand that there are many neurons that synapse with alpha motor neurons in the spinal cord that are not just primary motor cortex neurons. Okay? There are other neurons in the spinal cord too that synapse with these alpha motor neurons, and the neurons that we're going to talk about in particular are called GABAergic neurons. Okay, so there are GABAergic neurons in the spinal cord that synapse with alpha motor neurons, and what exactly do they do? So let's pretend that this is a GABAergic neuron, GABAergic neuron, and let's pretend that this is that alpha motor neuron in the spinal cord that goes out and synapses with skeletal muscle fibers, allows them to contract. All right, so what does this GABAergic neuron do? Well, when it generates an action potential, it passes that action potential down into its axon terminal buttons, just like any other neuron does. And what it releases from its axon terminal buttons is what's called gamma aminobutyric acid, GABA. Okay? They're called GABAergic neurons because they release GABA. Okay? And as you can see, as I'm trying to depict in this image, it's releasing GABA onto this alpha motor neuron. And what does GABA do to this alpha motor neuron? Well, located on the dendrites, you have to pretend that this is the dendrite, of that alpha motor neuron are GABA receptors. Okay? So this is the GABAergic neuron's axon terminal button, and it just generated an action potential, the GABAergic neuron. Okay? So sodium is flowing in to the axon terminal, making this region positive, flows into the axon terminal button, opens up voltage-gated calcium channels, calcium flows in, and then it causes these vesicles filled with GABA, gamma aminobutyric acid, to go to the distal end of the button, dock, and then GABA gets released into the synaptic cleft. Okay? And now GABA is going to bind to these GABA receptors located on the alpha motor neuron's dendrites. And it causes these GABA receptors, of course, to open. And what are these GABA receptors? They are channels for chloride calcium. I've gotten too used to saying sodium and calcium. So what's that going to allow for? First of all, chloride is a negative ion. Okay? Chloride is a negative ion. It's high in the extracellular fluid and very low in the intracellular fluid. So it has a chemical driving force to enter. Okay? But you might say, well, isn't the neuron negative 70 approximately, negative 70 millivolts? So doesn't the neuron, the alpha motor neuron in particular, not have an electrical driving force for it to enter? And the answer to that is yes, it doesn't. Chloride does not have an electrical driving force to come in to the neuron, but it does have a chemical driving force to come in. And here's what we know about chloride. Within neurons, chloride's equilibrium potential is like, it really varies from neuron to neuron, but it's more like negative 90 approximately. It can be even higher than that, even more negative, I mean. The point is to say that at this point, when a neuron is at negative 70 millivolts, chloride's chemical driving force to come in is still stronger than the electrical driving force pulling it out. So if GABA binds to GABA receptors, which are really, ligand-gated channels, which are, chloride pores channels on the dendrites of the alpha motor neuron. What GABA binding to these GABA receptors does is it increases the permeability that the dendrite has for chloride. And since chloride's chemical driving force is still stronger to come in than the electrical driving force pulling it back out, if you increase the permeability that the dendrite has for chloride by opening up these GABA receptors, which are chloride channels, then chloride is going to come in to the dendrites of the alpha motor neuron. Okay, so chloride is going to begin to come in to the alpha motor neuron. Back to this picture, if this GABAergic neuron is firing, generating action potentials, and releasing GABA from its axon terminal buttons, then GABA is binding to the GABA receptors on the dendrites of the alpha motor neuron. And those GABA receptors are chloride channels. So what the GABAergic neuron is doing to the alpha motor neuron is increasing the alpha motor neuron's permeability to chloride by GABA opening up these GABA receptors on the alpha motor neuron's dendrites. And then what chloride will do is it will enter into the alpha motor neuron because it has an increased permeability. And now the alpha motor neuron is gaining these negatively charged chloride ions. So what's going to happen to the alpha motor neuron is it's going to hyperpolarize. It's going to become even more negative. It's going to go from -70 to -75 or -80, right? It's going to become even more negative. The purpose of GABAergic neurons is to hyperpolarize other neurons. It's to make them more negative by allowing chloride into them. GABA sometimes has different effects in different regions of the nervous system, but this is true for alpha motor neurons in particular. GABAergic neurons hyperpolarize, make alpha motor neurons more negative, and they do it through chloride. So what do these GABAergic neurons in the spinal cord then do to the alpha motor neurons? Here's a GABAergic neuron. The GABAergic neurons make it harder for the primary motor cortex neurons to actually activate, get the alpha motor neurons to threshold. So the GABAergic neurons in the spinal cord kind of regulate, they have a huge role in regulating the activity of alpha motor neurons, okay? Because as long as this GABAergic neuron is firing, it's hyperpolarizing the alpha motor neuron to some degree, and it's making it harder on the primary motor cortex neuron to get the alpha motor neuron to threshold. Because what's the alpha motor neuron what's the primary motor cortex neuron going to be releasing? It's going to be releasing glutamate, right? So the primary motor cortex neuron is releasing glutamate onto the alpha motor neuron, and the GABAergic neuron is releasing GABA onto the alpha motor neuron. So they're fighting each other. Glutamate is going to be binding to glutamate receptors on the alpha motor neuron's dendrites, allowing sodium in, and GABA is going to be binding to GABA receptors on the alpha motor neuron's dendrites and allowing chloride in. So these two different neurons, GABAergic neurons and primary motor cortex neurons, are constantly battling each other to try to get the alpha motor neurons to threshold. If the primary motor cortex neurons win, basically what that means is they fire more frequently than the GABAergic neurons, then the alpha motor neuron is going to fire and the skeletal muscle fibers are going to contract. So how does all of this relate to tetanus? The bacteria that causes tetanus, what it does is it gets into the nervous system. In particular, it goes into GABAergic neurons and it knocks out what's called synaptobrevin. What is synaptobrevin? All right, so these are the vesicles within the axon terminal button of the GABAergic neuron. Okay? And these vesicles, they actually have to dock to the distal end of the button to release their neurotransmitters GABA into the synaptic cleft. And the way that these vesicles dock to the distal end of the button is by using different types of snare proteins. And one of them is synaptobrevin. Here is a vesicle, and you can imagine this vesicle is filled with GABA. Okay? And this vesicle, what it does is it has this snare protein that's called synaptobrevin. Okay? And it uses synaptobrevin, this snare protein, to bind to other snare proteins located on the plasma membrane that is the distal end of the button. So this would be like the distal end of the button here, okay? And the distal end of the button has snare proteins too. And those snare proteins bind to the snare proteins located on the vesicle that has GABA within it. So you can see the distal, like the button, the distal end of the button, its snare proteins bind to the snare proteins, in this case synaptobrevin, located on the vesicle, in this case that houses GABA. Another image here, okay, here, I'll do this in red. Here is the vesicle. Inside is GABA. Okay? This vesicle has these snare proteins. One of them here is synaptobrevin. Okay, this snare protein, synaptobrevin, is going to bind to other snare proteins located on the plasma membrane, that is the distal end of the button, of the, of the axon terminal button. Okay, what the bacteria that causes tetanus does is it gets into GABAergic neurons and it knocks out synaptobrevin, this snare protein. If synaptobrevin is knocked out, this snare protein associated with the vesicle, then the vesicle can't actually dock to the distal end of the button, and then GABA cannot be released from the GABAergic neurons. As you probably have figured out with me at this point, I like to show lots of different images of the same thing. Here you can see one more time, this is the vesicle. Inside of the vesicle, there's GABA. Okay. This in red, right here, I'm drawing it in red, is synaptobrevin. Okay. This snare protein that's associated with the vesicle that houses GABA. And this is a depiction of the distal end of the button. Okay. That vesicle needs to dock to the distal end of the button so that GABA can be released through exocytosis out into the synaptic cleft. Here you can see synaptobrevin binding to the other snare proteins that are associated with the distal end of the button. If the bacteria that causes tetanus gets into the GABAergic neurons and knocks out synaptobrevin, then the vesicles cannot bind to the distal end of the button and GABA cannot be released from the GABAergic neurons. If GABA cannot be released from the GABAergic neurons, right, because synaptobrevin has been knocked out and the vesicles can't bind to the distal end of the button, then GABA cannot be released into the synaptic cleft, and GABA will not bind to the GABA receptors on the dendrites of the alpha motor neuron. So what tetanus does is it knocks out, essentially, these GABAergic neurons. Right? It prevents these GABAergic neurons from actually being able to release GABA onto the alpha motor neurons. And what happens is the alpha motor neurons then become incredibly hyperactive, and thus the skeletal muscle fibers, which they innervate, right, end up contracting uncontrollably, right? And you get these crazy spasms. These muscle fibers end up in a state of tetanus, maximal contraction in terms of force, and death can occur. And in most cases, it does occur. Luckily, though, there are very effective vaccines against tetanus. They're quite effective. The studies vary, but it's somewhere between 80 and 95% effective. Most children that get the tetanus vaccine are very well protected against tetanus. Next, we will talk about a interestingly very popular bacteria which is the botulinum bacteria, which can cause botulism. Now, the mechanism of action for botulinum toxins is very similar to that of the bacteria that causes tetanus, except the botulinum toxin goes after a different neuron. Botulinum toxins, go after alpha motor neurons. Okay, they get into the axon terminal buttons of alpha motor neurons, these neurons that release acetylcholine. And botulinum toxin gets into these alpha motor neurons, axon terminal buttons, and from there it's very similar to what the bacteria that causes tetanus does to GABAergic neurons. There are different types of botulinum toxins. There's, first of all, B, D, F, and G. What they do is they get into the axon terminal button of the alpha motor neurons and knock out synaptobrevin, just like the bacteria that causes tetanus. So you can imagine this as the vesicle inside of the alpha motor neuron that has a CH within it. Okay? And if the botulinum toxin gets into this axon terminal button, what it will do is it will knock out synaptobrevin, and that will prevent the vesicle that houses acetylcholine from being able to dock to the distal end of the button, and ACH will not be able to be released. Now there's also botulinum AEC1, and what they mostly do is they knock out the snare protein, that is SNAP25. Okay, so they don't knock out the snare protein synaptobrevin, they knock out the snare protein SNAP25. Now it's going to result in the exact same thing, but let's show you that. SNAP25 is a snare protein that is located in the distal end of the button. It's located in the plasma membrane of the axon terminal button. Okay, but it's another snare protein. Okay, if this snare protein within the distal end of the button is knocked out by botulinum toxin, then again, the vesicles that house acetylcholine are not going to be able to dock to the distal end of the button, and acetylcholine will not be able to be released into the synaptic cleft. Okay, so botulinum toxins and the bacteria that causes tetanus really function the same way. Their mechanisms of action are very, very similar. But they go after different types of neurons, and thus their ultimate effect on the human body is going to be the complete opposite. So the bacteria that causes tetanus, it goes after GABAergic neurons. It enters into GABAergic neurons, interferes with the snare proteins in their axon terminal buttons, and thus prevents them from releasing GABA. And then the alpha motor neurons become hyperactive. Now, the botulinum toxin enters into alpha motor neurons, not GABAergic neurons. It enters into alpha motor neurons, and what it does inside of the alpha motor neurons, again, in terms of mechanism, it's the same thing as the bacteria that causes tetanus. It goes into the alpha motor neurons and it interferes with the snare proteins that allow for the release of the neurotransmitters. But in this case, it's preventing the release of acetylcholine, because that's what the alpha motor neurons release. And if you prevent the release of acetylcholine, that's what the botulinum toxin does, then action potentials won't be able to be passed to skeletal muscle fibers, and those skeletal muscle fibers will thus be put into a state of paralysis. They'll be paralyzed. The botulinum toxin, what it does is it prevents skeletal muscle fibers from being able to contract. And it does that by preventing alpha motor neurons from being able to release their acetylcholine via exocytosis, which means skeletal muscle fibers cannot receive action potentials and thus they cannot contract. So let's just think about this real quick. The tetanus bacteria gets into GABAergic neurons and prevents the GABAergic neurons from releasing GABA. The botulinum toxin gets into alpha motor neurons and prevents the alpha motor neurons from being able to release acetylcholine. So the tetanus bacteria and the botulinum bacteria, right, have the same mechanism of action, but they target different neurons. So the tetanus bacteria knocks out GABAergic neurons and thus results in alpha motor neurons being hyperstimulated and muscle fibers, skeletal muscle fibers, overly contracting, right, muscle spasms, which can result in death. The botulinum toxin, what it does is it prevents alpha motor neurons from releasing acetylcholine, and thus skeletal muscle fibers can't receive action potentials, and thus the skeletal muscle fibers don't contract. They're put into a state of paralysis, which also can result in death. The botulinum toxin is actually one of the most potent toxins that exists. You don't need much of it for it to be fatal. So interestingly, the tetanus bacteria, the botulinum bacteria, they both can kill people, right? But the tetanus bacteria does it by causing skeletal muscle fibers to contract way too frequently and forcefully, spasm, and the botulinum bacteria does it by not allowing those muscle fibers to contract at all. And now this bacteria is actually used, as most people are familiar with. I'm not saying most people go out and get Botox injections, but it's actually fairly common. Botox is the botulinum toxin, and it's injected typically into the face to prevent contraction of muscles within the face by knocking out the alpha motor neurons that release acetylcholine onto those muscle fibers within those muscles, and thus it causes paralysis of these facial muscles. That, of course, can remove the appearance of wrinkles and other, I guess, less desirable facial features. Now, the body can slowly degrade the botulinum toxin, so I'm no expert on Botox, but you do have to frequently go back to get more injections of it if you want it to continue to give you the desired outcome, whatever that might be. Now, beyond purely cosmetic purposes, Botox is also used in some people with migraines. Migraines as a whole is a pretty mysterious phenomenon, but in some people, some clinical trials have shown Botox to be decently effective with migraines. This is an image showing the injection sites for those who get Botox for migraines. And people with diseases that result in muscle spasms. It has definitely shown to be pretty effective at assisting with making certain diseases that cause muscle spasms to be more tolerable. So it definitely has clinical benefit beyond just cosmetic purposes. Not that there's anything wrong with the cosmetic reasons for getting it, I'm just not as familiar with it.